So today I'm going to speak about NGS and its related challenges. And as usual, I will try to present concepts in a vulgarized way to simplify and raise awareness of the next generation of researchers to the next generation sequencing and its related challenges. So let's start with a brief history that illustrates the important milestones of the DNA sequencing story. I guess that many of you know that the discovery of DNA dates back to 1870. However, the remarkable achievements dates back to 1953, when Watson and Crick discovered the double helix structure of the DNA molecule, and this discovery paved the way to many molecular biology applications. Maybe the most important was the sequencing, which was performed on bacterial RNA the first time. And it's only in 1977 that we started speaking about Sanger sequencing, which is the gold standard technique and the only direct genotyping method so far available. So Sanger sequencing is still used today to confirm any findings identified by all indirect genotyping methods and even NGS. So this technique is developed by Sanger, here shown in the photo, and is based on the differential termination of the DNA fragments by incorporating modified nucleotides that inhibit the prolongation of DNA synthesis, and these nucleotides are labeled either by radioactivity first, when the technique first started, or by fluorescence, which is used nowadays. So in 1990, when cycle sequencing has been introduced as well as automation, a big project that has marked the genomic history has been launched. And this project is the Human Genome Project, which has been launched by the President of the US at that time, Bill Clinton, and next to him, Francis Collins, who is the present director of the National Institutes of Health. And the Human Genome Project was also launched by the Department of Defense, and around 2,500 scientists were involved in this project from 20 research institutions and spread over 6 countries, and they were involved for 13 years of work. And in 2003, they give birth to the first complete human genome sequence. So this first project and this first sequence of human genome cost the humanity the price of $3.8 billion, which is a huge amount. So after unraveling this first sequence of this molecule, the attention has been paid how to get this sequence a second time, but cheaply and quickly. And this goal has been made possible with the introduction of next generation sequencing in 2006. So let's have first a little brief overview about this technique. So NGS, which is called also high throughput sequencing, it uses a number of modern technologies, and it allows sequencing of both DNA and RNA rapidly and cheaply, relatively cheaply compared to Sanger sequencing. And this technique has really revolutionized our understanding of genetics and molecular biology, and also our comprehension of genetic diseases mechanisms. So from a technical point of view, NGS is performed through 3 steps. The first one consists of digestion of DNA, which is followed by its amplification, as shown here in this video, which is done on a flow cell and which shows bridge PCR and formation then of different clusters. Each cluster like this one represents a unique DNA sequence. And I would like to thank my colleague, Dr. Yusra Bouyakoub, from whom I borrowed this slide and the next slide. So the amplified fragments are then automatically sequenced, and we call this technique the sequencing by synthesis, because each time we will have a nucleotide incorporated, we will have a fluorescence which is emitted here and which is captured by the machine and then translated into letter to form at the end the whole sequenced DNA fragment. This slide is just to give you an idea about the difference between Sanger sequencing, electropherograms here, and NGS, the different reads, and how to assign heterozygosity for both techniques. So since that time, so since 2006, competition has reached its peak between companies to offer the best. And the third generation of sequencing machines was set up, which is based on other sequencing principles, like, for example, here, Pacific Bioscience, which uses the nanopore technology and offers long reads sequencing. And of course, those are helpful for other applications. At the end of this DNA sequencing road, I want to cite HiSeq. Even though the road is still long, we have others like NovaSeq, which are present here on the market. And but why I have chosen HiSeq? Because its sequencing capacities reach another scale of performance. And to give you an idea with this slide, the evolution of techniques, and try to compare between the efficiency and the performance between Sanger sequencing and NGS and HiSeq, and I think the numbers speak by themselves. So, sorry. So with these technological advancements, the price of NGS is getting lower and lower, and this affordability of this technique has permitted scientists to launch big projects like the one which is very famous, which is 1,000 genomes, which started in 2008 and finished his pilot phase in 2010. And after that, other initiatives in many countries are continuously achieved, like in 2017 in specific populations, and more recently, and others studies to sequence individuals. Here, I would like to draw your attention to this figure on the right, which shows a consistency between the sequencing performances and computing performances. And this represents the first challenge that face NGS. So now I will try to list some challenges that are facing NGS from my point of view, which is the goal of my talk. So as I said, this is an automated technique. And when we speak about automation, we speak about machine-based interpretation. For example, here in the flow cell, which is the device used to sequence to sequence the DNA, let's say, the fluorescence is not always in the optimal condition like this, but the signals obtained and read by the machine may be sometimes fuzzy or overlapping like this, or sometimes even absent. And this creates a lot of false negative and false positive results, and sometimes even missing data. Here, also another example, the software, which will assign each read to its reference human sequence, it's not easy as a task, since sometimes we have many similar regions and repetitive elements. You know, our genome is rich of repetitive elements, and the software sometimes find many difficulties or ambiguity doing this step. So let's move now from the technical issues to speak about money. And here I give you some numbers relative to cost estimation for a NovaSeq. It's an example of a machine for sequencing. And this NGS machine has the capacity to produce 24 genomes per day. So that means in a year, it is able to produce 9,000 genomes. So if we tell you that one genome costs $1,000, that means if you use this machine at its maximum yield, you will have to pay for reagents, only for reagents, $24,000 every day, and more than $8 million per year, which is really a huge amount. Okay, let's imagine that you have the money and you are working in a rich lab, and you are able to sequence and use these machines at their maximum yield. Sequencing will, of course, generate data, and it will generate a huge amount of data. So to illustrate this data gathered by one HiSeq run, it needs about 2,097 CDs to be placed, to store this data. And if we put these CDs on top of each other, we will have a height of 2.5 meters. So fortunately, we don't store NGS data in CDs. And to overcome this problem of data storage, One of the alternatives is clouds, and nowadays many clouds are available to store biological and more precisely genomic data, as shown here. But keep in mind, there is nothing for free. You have to pay also. So the cost of paying storing data on cloud is 25 centimes per gigabyte per month. But we don't have gigabyte data. We have 6 terabyte per day production by this machine, are produced by this machine every day. And so if we calculate the amount, it will be around $675,000 per year. Don't forget that this amount has to be added to the 8 million we have already paid, or we have to pay to generate the data. And I don't think, honestly, that our labs can offer such services. So here, just to give you an idea what is petabyte and what is cetabyte, this give you an idea. So an exabyte takes up a city block, and the zettabyte is almost half of Bahrain surface also. And yottabyte is equal to the surface of Lebanon. So it's really a big and a huge amount of data, and we have to think on solutions for data storage. So let's now move from money to the scientific side, and let's suggest that we are working on rare disease, which is my specialty. So for rare disease, we will have to look for rare variants, and the causative variant should segregate with the disease, should affect protein structure and or function, and should be absent in healthy individuals, let's say at least in his family or relatives, and should also have a very low frequency in the general population. Here, for this last point, I would like to emphasize on the fact that, unfortunately, our populations, and I mean by our population, the Arab world, we are not well represented, if not yet absent, from the available public genomic databases. So be careful, especially if you want to look for rare variants. And so looking for the variant behind the disease is like looking for a needle in a haystack, but the needle is not clear that way. I represented here in the figure. You have instead to have a closer look at the sequencing results while being well equipped with the necessary bioinformatics tools to examine the data. And keep in mind that in the majority of cases, the situation is not clear like this photo, but it is very confusing like that. Another challenging is facing our understanding of genomes is our limited understanding of genome. Since the coding sequence accounts only for 1 to 2% of the genome composition, so if we do a whole genome sequencing, of course we can read the sequences, but we cannot interpret 98% of our genome, which is a huge proportion. And why this is important? Because this represents the primary challenge of whole genome sequencing and coding variant interpretation. And this limitation can explain why, so far, we have identified only 450 strongly pathogenic human regulatory variants in the non-coding regions. However, we know more than 2,100, I think, variants in the coding regions. And one of the plausible explanations is the difficulty to confirm their pathogenic effect. So here I want to say that the genome has to be interpreted not as a linear sequence, but as a 3D structure to assess potential distal actions. And I would like also to add that we should not forget the part of epigenetics, as already shown in the presentation of my colleague Shamsuddin, and the impact of the environment on the variable expressivity between individuals. This is why we cannot accurately predict the risk for a given disorder. Here we can take the example of twins who are, for example, identical, but we cannot affirm the predisposition to a given disorder, even if one of them expressed the disease. And this is really true and mainly valid for complex diseases. And why for complex diseases? Because, as I already said, identifying the causative variant should be confirmed by functional studies. We should combine all these components, genetic and environmental and epigenetics, to correlate the clinic to the genetics. So we need functional validation to correlate what we observe as a phenotype to what we observe as a genotype and understand the pathophysiology behind the disease. And this step is very important and very crucial as it paves the way for all the process from therapeutic design to clinical trials. So even if the functional effect is confirmed, the genetic background should be analyzed in a larger context that includes, first, a structured phenotyping with a very good clinical description. And second, an understanding of a very profound genetic description or genotyping that takes into account all the modifier genes, the whole genome, and epigenome signature. Third, an understanding of the normal physiology. And this, of course, will lead to a fine and precise diagnosis to better define the appropriate management. Here, I would like to illustrate this complexity by a recent example taken from the coronavirus epidemic. Indeed, we all agree that NGS was a powerful tool to decipher the genomes of the viruses very quickly, and this was helpful for setting up the detection kits and used in diagnosis and to control, of course, the propagation of the disease. But the ongoing work now is to continue working on these sequences and to combine the viral genomic sequence with data from lung microbiome and gut microbiome as well, together with the genetic background of the individual, not of the viral, of the host, as crucial elements together to understand this host pathogenic interaction and to better evaluate the prognosis of the disease. And I am sure that this would lead certainly to a precise treatment conception. So now another layer of complexity corresponds to one of the applications of NGS, which is metagenomic sequencing. Here we are dealing with a mixed sample. So the main issue is how to assign each sequence to each species. In such metagenomic applications, there is one problem which is very important, which is the problem of unmapped reads, which are mainly due to the non-availability of the reference genome sequence, and here the possibility, of course, of discovering new species arises. So let's move to another aspect. So working with biological material, and especially human DNA, implies many ethical challenges that are mainly related to incidental findings for future onset disease. Let's give an example to illustrate this concept. The example of, for example, a pre-symptomatic patient who is an individual for whom we have identified a disease-causative variant, but the person is still normal, he is healthy. Here we suggest that if he lives longer, he will develop a cancer or a neurological disease or anything else. So from a genetic counseling perspective, do we tell him to start preparing his life post-retirement? Maybe the disease will start in the age of 80 and he will die before that age. This is the case, of course, of pre-symptomatic individuals, but also for patients' relative and cascade screening and for variant of unknown significance. So the main question is, what is the guidance for an appropriate genetic counseling? So now, after listing many challenges related to NGS applications, I would like to bring you with me into a future projection and discuss possible solutions or alternatives. So if we start with the technical side, I think that the technology will continue to advance, but the question is, are we really ready to these advancements in terms of data storage, result interpretation, the legal framework? So the first solution is to find a way to be rich and to have the necessary money to pay your whole genome. And for this, you are, and all of us are highly encouraged to establish international collaboration and work in big consortia. The second solution is you have to be a good biologist and do not forget the fundamentals of genetics and molecular biology and physiology. Because remember that the process is totally automated and is based on machine analysis. So we need the eye and the mind of a biologist. This is why, for example, here I give an example of GeneDx, which is the company #1 in the world, in the US, in genetic diagnosis. It hires thousands and thousands of biologists who are doing the same job, genetic analyzer. So let's hear I want to give a message of hope for all the biologists who are jobless, and unfortunately there are many in my country. It is estimated that by 2030, this job, which is called genetic analyzer, will exceed by far all jobs already known and will know a demand never known for all existing jobs, we hope. So for problems related to data storage, there is an alternative, which is the 5D optical data storage. So this memory crystal can store up to 360 terabytes of data for billions of years. And another solution is DNA. Yes, DNA. DNA is used to store huge amounts of data, and it could be the future of the data storage. And it's also a robust tool. And the process is just based on converting the binary code of any file you have to a nucleobase code and vice versa. This solution seems very promising since many companies have started using it, and it can store all the data, in all the world's data as shown here, in just 1 kilogram of DNA, which is really something incredible.
Sorry, Olfa, but I think that the time is already up. So
I will finish with this slide.
Okay.
Yes, I will finish now. So, and finally, of course, I will finish with ethical and regulatory framework. So, here I want to give you some questions just as a food for thought. Do we import laws from outside? And if so, are they applicable to our cultural context? If not, do we generate our own laws? And can we speak about internationalization of laws and rules that regulate genetics and genomics? And please, finally, keep in mind what several companies are doing now, like Family Tree and 23andMe, that offer ancestrally personal genetic services. Is it harmless to collect genetic data on human beings? Please think about population segregation. Think about genetic war, and thank you. And at the end, I would like to thank my colleague, Dr. Ahmed Shakroun, who is the founder of RunBiolinks, a company for genetic services, who helped me in elaborating some of the ideas presented here. And thank you all for your attention. Happy to take questions. Thank you, Shamsi.